Okay. So, hello everyone. I hope you guys are having a good weekend and you guys are doing well. I hope so. So I am Chandra, and I have been like helping you guys with this with some problems of this course for the past now 7 weeks. So next week will be the last week for the sessions. So those of you who have who are joining for the first time, a brief introduction myself will help you know more about me. So I'm actually a PhD scholar in the Department of Chemical Engineering at IIT Kanpur, and I'm working in the field of tissue engineering. If you like to know more about my work, I have given the link of my website. Feel free to go and check it out. Every Saturdays we used to meet earlier from 4 to 6, but now I've shifted the time to 6 to 8. So please note the meeting link, everything will remain same. The recordings will be available in my YouTube channel, and the notes are available as well. So for the meeting, you won't be able to use the chat option. Feel free to unmute yourself and ask any queries. So quickly, I'll recap some of the topics discussed in week 7 before I go ahead and we solve few problems. So first we saw the basics of immunology, basically, like what are the cells of immune system, right? Then what are the organs? Then we got familiarized with some um, terminologies like adaptive and then innate immunity. Then we also got to know what is cell mediated immunity and humoral immunity, right? So these are pretty basics. You guys must have studied them in your immunology course. So it was just a primer of that. Next, the crucial topic for tissue engineering is this one. We understood how does the body react or respond to biomaterials or tissue engineered products after implantation. All right. And we saw something known as foreign body reaction. So let me quickly go through how is the process. Okay, how does the process goes on in our body. So if we visualize it in form of a time scale like this. So the moment you are implanting your tissue engineered product or a biomaterial device or something like that, what you are doing, you're creating an injury, right? So it maybe you are like simply inserting it under the skin, or you may be injecting it, making a very tiny injury, or it may be like you have to create a surgical insertion and then implant it. So whatever you're doing, you're basically penetrating the body's primary defense system, that is our skin. And then the deeper you go, you will puncture the inner organs as well. So what you are doing, you are creating an injury. So when the injury is made, the first thing that our biomaterial will get in touch with is blood. And blood contains a lot of other things like proteins, different type of cells, right? So all those things get in contact with our biomaterial within few seconds, all right, within few seconds to minutes. And blood material interaction goes on. Right? So during this time, what happens is that whatever proteins are there within our plasma, they get absorbed on the surface of your biomaterial and creates something called as, so let me, so this is like this entire region, let's say, is the blood material, interaction panel, and here blood gets in touch with your biomaterial and provisional matrix is formed. Okay, so it is nothing but protein adsorbed on the surface of your biomaterial. Okay, so it also creates a surface onto which your immune cells like neutrophils and all, neutrophils, then platelets, they can come and adhere onto it. So, and this provisional matrix, apart from the fibrinogen is a protein that is found in our plasma that gets cross-linked and forms fibrin. So this provisional matrix is primarily a fibrin matrix into which cells like platelets, neutrophils, and other cell debris, they get trapped. So once that happens, these cells, they are now localized, right, onto the surface of a biomaterial. What they will do, they will start secreting cytokines or different kinds of chemoattractants in order to call more cells at the site of the injury. So, so when that happens, then begins the inflammation process. So, why inflammation happens? So, inflammation basically tries to contain the damage within the local area. Since the barrier is penetrated, so any microbe or any other foreign must have entered into our body, bacteria and all. So our body tries to make sure those things doesn't penetrate further deeper inside the body. So it tries to elicit as much harsh response through secretion of proteases, then ROS, reactive oxygen species. They are the most damaging substances released by the inflammatory cells, and they try to kill those foreign particles like the biomaterial and the associated microbes and all. So that it doesn't create any more damage further. That's the inflammation. So if the inflammation if it can last for a very short period of time, so when that happens, it is called acute inflammation. And if the process of inflammation goes on for relatively higher time scale, like for months or years, then it will be called chronic inflammation. Okay. Generally, acute inflammation is desired. Okay. So initially, as I told you, neutrophils are the ones to come. Now, these neutrophils, they secrete cytokines and call in monocytes at the site of the injury. Now, these monocytes, they differentiate and macrophages also arrive here. So these are phagocytic cells. Their main job is to phagocytose. So phagocytosis happens in three stages. First, it will recognize the foreign thing. Okay. Then it will engulf it, and then through release of some hydrolytic enzymes, it will destroy it. That is the three steps phagocytosis that is done by these cells and monocytes. Now, in case of biomaterials, phagocytosis is not possible because its size is generally larger than the monocytes size or macrophages size. That's why um, these cells, they tend to stay near to the biomaterials because they cannot engulf it and show a response known as frustrated phagocytosis, where it releases more and more proteases and ROSs to destroy this material, and it's like a very vigorous response goes on. Anything smaller than 10 micron and all, it can be easily engulfed. But larger sizes, like the biomaterial devices and the tissue unit products, which are generally larger sizes, for them, phagocytosis is not possible. Now, if suppose the threat is not, say, neutralized during the acute inflammation step, then it will continue towards chronic inflammation. Okay, when the same thing goes on, but for a larger time scale. All right. Now, as the inflammation is happening, then also starts the formation of granulation tissue. So these are like overlapping events now. Now, at this stage, if suppose inflammation is going on, then what happens? The remaining tissues within the remaining cells within the tissue, meaning functional cells in the tissue, which are damaged, like the endothelial cells, then fibroblast, they start proliferating. Okay, they starts proliferating because that wound is still open. We have to close it. We have to deposit enough proteins and ECM components to make it mechanically rigid, so that is like it is not susceptible to any more damage. So for that, these granulation tissue is formed by these cells, endothelial cells and fibroblasts, and they also secrete ECM components like collagen, proteoglycan, etc. Okay, so a granulation tissue looks like very pink, very translucent kind of a matrix, and it has a granular appearance, thus the name granulation tissue. Now, after granulation tissue is like almost to the completion, then starts the FBR or foreign body reaction. Now, as I told you, right, the biomaterial is a big substance, right? It's larger than the size of the monocytes and macrophages. The inflammation is still going on, by the way, okay? Like granulation tissue, although it has like almost formed, but inflammation is still going on. And it has reached to a stage such that of FBR has started, foreign body reaction has started. Now, at this stage, the macrophages now they have fused. Okay, they have fused to make something called foreign body giant cells, FBGC. So, a large number of macrophages they fuse together and create very big cells in order to compete with the size of the biomaterial itself. Now, this response is again the same thing, like they releases large amount of proteases and the ROS and to attack and to degrade the biomaterial. Okay. Now, if the material is like inert and not inert, if the material is not at all biodegradable in nature and will not at all respond to these foreign body giant cells, then what people have seen is these cells they tend to like stick to it. Okay, just they are just there. And when people have removed those transplants from the body and done ex vivo analysis, they see those cells still attached to it. Okay, so but they are kind of a dormant stage, like resting stage kind of. So I don't know, people have not reported like how much extent it is still going to do, especially for materials which are of non-biodegradable nature. But yeah, that is what is called FBR, that is mediated by the foreign body giant cells, and they still try to phagocytose the biomaterial itself. Now, the as you can realize that these ROS and the proteases, they are very harsh, right? They can also affect the surrounding healthy tissue. So, in order to contain that, our body like have a very nice way of like encapsulating it. It happens through the formation of fibrotic capsule or fibrosis, it is called. It is called fibrosis because large amount of collagen gets secreted and it basically creates a capsule around it. Now this fibrotic tissue, it itself has no function. It's like a large amount of scarred tissue, and it does not mimic any of the tissue properties. So but its only job is to contain the foreign body reaction that is going on, so that the surrounding tissue remains healthy. So after this fibrosis happens, many a times the device, which you have implanted, it can stop working, because there is now like almost no flow of nutrients or oxygen or removal of waste material. So whatever you have transplanted, it may stop functioning if it has healthy cells in it. But if it's like a sensor based thing, like a chip and all, I think it can still work. But I don't know. I've not worked with these kind of I've not worked with fibrosis as a topic yet. So yeah, so these are the steps, that ultimate fate of any biomaterial that goes into your body. It's foreign, so it undergoes fibrosis. It's a very good research area actually. People are trying to negate this effect, like deliver some anti-fibrotic agents so that the fibrosis doesn't happen, and their delivered cells, the tissue engineered products, they remain healthy and they can do their job for a longer period of time. Okay, so this is a very new area of research, and many work is still yet to happen in order to, either induce fibrosis if it is needed or restrict it. So, yeah, this is a general overview of how our body responds to a foreign biomaterial. So, um, I hope you have, like if you have gone through the lecture, it might have helped you to recall it. If not, I think it might help you to, understand better later when you watch the lectures. Now, next topic that was discussed is bioethics. So it's not very difficult to understand, right? So it's basically depends on what you think is ethically correct and not correct. So there are committees in different institutes that our ethics committee. They try to see the value of your research. If you are working with the animals, then they will ask you how research is going to means is your research worth sacrificing life. So these kind of questions people ask, and you have to also question yourself, how much are you willing to um, like harm other living organisms for the benefit of research. So go through the lectures. These are very small lectures uploaded in the NPTEL. So I don't want to get more into it because I find the lectures very boring, to be very honest. But yeah, please go through it, and you'll understand it is fairly easy. And the next topic that we saw this week is about skin tissue engineering. So here we discussed about skin anatomy, right? How the structure of the skin looks like, because that is the first and foremost thing that you should know when you're targeting a organ for tissue engineering purposes. How the organ looks like, what are the components, how many layers are there, how many cell types are there. Okay, so what are its functions, so you should know that. And then some attempts for tissue engineering in skin, for skin, has been also discussed, like membrane based approach, then cell-seeded membranes as a tissue engineered products. So these things have been discussed, how they are designed, what are the criteria for designing, what are the challenges for transplantation. So these things are discussed. Please, again, I request you to go through the lectures, and you will get a better idea. So, so far, if you have any queries, please let me know. I'll be happy to take it up, and then I'll move on to some problems. Yes. Any queries? All right. Let's move on to some questions. Um, are you guys here in the meeting? It would be good to know so that yeah. Okay. Thank you, Avinash, for confirming. Okay. So let's start with the questions. The first question: what type of macrophages are found in the connective tissue? So, from the basics of immunology, that there are some macrophages who migrate out from the bloodstream and go and reside to some tissues. They are called tissue specific macrophages. And although they are natively macrophages, but they are named differently. Okay. So, in different organs, they are found. So, these are the names of those macrophages. You have to recognize which of these macrophages are found in the connective tissue. So, option A is histocyte, B is microglia, C is Kupffer cells, and D is mesenglial cells. Yes. What do you think is the correct answer? If you have gone through the lectures. If not, it's fine. Okay. So the correct answer here is histocytes. Histocytes are the macrophages that are found in the connective tissue, whereas the microglia is found in central nervous system. Kupffer cells are found in liver. Mesenglial cells, they are found in kidney. There's one more type of cells called Langerhans cells. These are also macrophages and they are found in skin. Okay, just a general knowledge a bit. This is the next question. What is the primary function of immune system? A, to digest food, B, to digest hormones, C, to protect the body against infections, and D, to regulate the body temperature. Yes, go ahead. You don't have to raise your hand, unmute, and yeah, go ahead.
Option C, to
Yeah,
protect the body against infections.
yes, you're correct. You got it right. So it's C. This is the next question. Which of the following is the part of body's first line of defense in the immune system? A. Antibodies, B. Skin, C. White blood cells, D. T cells. The first line of defense is talking about the innate components of innate immunity, basically the barriers. Okay. So which of the following is the body's first line of defense or the barrier of our body? A. Antibodies, B. Skin, C. White blood cells, T and D is T cells.
Antibodies.
Mm, it's actually skin. Okay. So antibodies they come later. Like they are not immediately there. So it's the skin that prevents the entry of any foreign objects into your body. Okay. If suppose that barrier, like if the skin is compromised, then once it enters inside your body, then the work of other cells like neutrophils, monocytes, they try to kill it off, phagocytose it. If that's not working, then um, antibodies get synthesized by B cells, and then T cells also help in this process by presenting these antigens to B cells, and specific antibodies get synthesized. So yeah, so it's a delayed response. Any response by T cells and antibodies, they are delayed. Whereas white blood cells, among the white blood cells, the neutrophils and the monocytes, they are the first one any foreign object encounters. But anything prior to that has to first go through our skin barrier. Other than skin, there is also the openings, right? The nasal passages are there. Then stomach lining is there. Whatever you eat, it goes inside your stomach, right? So there is acidic pH. Then mucosal lining is also there. So these mucosal membranes, the acidic pH in the stomach, our skin, these are the first line of defense that the microbe has to first pass, or any foreign object has to first pass. Okay, this is the next question. What type of cells they are primarily responsible for producing antibodies? T cells, B cells, macrophages, and red blood cells. Yes, which of these cells are known to produce antibodies?
B cells.
Yes, it's the B cells. This is the next question. Which of the following describes the adaptive immune system? Okay, adaptive immune system. A. It provides immediate response to infections. B. It is specific to particular pathogens. C. It relies on physical barriers like skin. D. It is unable to remember past infections.
Is it option B? I am doubtful.
Yeah, it's option B. You're correct. So adaptive immunity basically comprises of your B cells, T cells, and antibodies. And the specific characteristic of adaptive immunity include uh, it can recognize a diverse range of antigens. So diverse, it is very diversified. Okay, and also it gives a very specific response that is tailored for only that specific type of antigen. And through the help of T cells, so there are memory T cells. This adaptive immunity can also remember, so it can have a memory of any antigen that has entered the body prior, so that if it enters again, it will give a much more harsher um, response to it. Okay, and it will kill it quicker. And it has the ability to recognize cells, a self from non-self. So when this particular property is compromised in someone, then the diseases, a class of diseases called autoimmune diseases starts. Okay, like rheumatoid arthritis and all. So when the immune system is unable to recognize that, or unable to realize that it is killing our body's own cells. Okay, so yeah, The correct answer is option B. Histamine, kinins, bradykinin, prostaglandins. They are the major mediators involved in the inflammatory response. Is it true or false? Yes. Have you ever heard of these um, chemicals before? Okay, that's fine. So the correct option is true. So this is the slide that discusses the role of these chemicals, and it will, if you go through this slide, you will realize why this is true. So when some injury has happened, the injured cells, they start secreting an alarm that yes, injury has happened, come and help me, and help me close this wound, right? So what are those chemicals? So the chemicals are called histamine, prostaglandins, kinins, and bradykinins. So when these chemicals are released as a signal, then some immediate responses happen. First, the blood vessels, they dilate in their diameter. That causes an increased blood flow. Okay, and also the permeability of the blood walls, blood vessel walls, they also increases. So that allows exudation of blood cells and plasma at the injury site. So in this way, primary immune cells like neutrophils and other type of phagocytic cells, they can easily go and arrive at the site of injury. So when this happens, then the inflammatory response begins. Okay. So yeah, if you have any queries at any point of time, feel free to unmute yourself and ask. All right. This is the next question. Which organ is known as the graveyard of red blood cells and also plays a role in the immune system? A. Liver, B. Heart, C. Spleen, and D. Lymph node. Which of the following is known as the graveyard of red blood cells and is also important for immune system? Actually, it is one of the organs of immune system.
Is it spleen?
Yes, you're correct. It is spleen. So spleen is where the red blood cells whose life is up, they go and they die there. But spleen is also the place where subsequent phagocytic activities by immune cells takes place. So if I recall correctly, so there are some organs, primary organs of the immune system, like bone marrow and thymus, where the B cells and T cells are formed. So these are bone marrow. They are like formed as well as they mature there. Once they comes into circulation and now active, there is a secondary site as well. So these B cells and T cells go to those secondary sites. These are usually your lymph nodes and spleen. At these secondary sites, the antigen, the foreign particles, which is phagocytosis and all, so they are now brought in. And here the B cells and T cells, they act on it and maybe synthesize specific antibodies to it, and register it to memory, and all those things happens. Okay, so these are the secondary site of action, and this is the primary site. Okay, so spleen is also the site where red blood cells die. What is the main difference between humoral and cell mediated immunity? What is the difference between these two types of immunity? Humoral immunity relies on antibodies, while cell mediated immunity, it involves T cells. B, humoral immunity is faster than cell mediated immunity. C, cell mediated immunity involves B cells, while humoral immunity involves T cells. And D, cell mediated immunity only targets bacteria, while humoral immunity targets viruses. So, which of these options are correct? That correctly describes the difference between humoral and cell mediated immunity.
Is it option C?
Um, it is actually option A. So humoral immunity basically talks about the use of antibodies and proteins. Okay, whereas cell-mediated immunity is actually a term that covers all the cell types, whichever cell types are involved in immune response. Okay, so although in this option it is written T cells, but it will also include the natural killer cells, the granulocytes and macrophages, monocytes and all. Okay, so yeah. What is the next question? Which part of the lymphatic system do T cells undergo maturation? Bone marrow, spleen, lymph nodes, thymus.
Is it A or D? I'm going for D.
Okay, you're correct. It is D. Okay. So at bone marrow, the B cells get matured, whereas T cells go to thymus for further maturation. But T cells are formed at bone marrow, but for maturation, it goes to thymus. This is the next question. What is the role of the complement system in the immune response? So yeah, complement system also comes under humoral immunity. Okay. So the role of complement system, which of these options is correct for it? A, to inhibit the activation of T cells. B, to stimulate the release of histamine. C, to enhance the ability of antibodies and phagocytic cells to clear pathogens. D, to prevent the immune system from recognizing self antigens.
Is it option D?
Why would it we don't we so immune system generally doesn't need to recognize the self antigens, right? So the option says that it will prevent the immune system recognizing self antigens. So it generally does not happen. So there is no need to prevent it. Maybe to give it another
Yeah,
try.
option C.
Option C, yes, it's correct. So as the name suggests, complement system. So its role is to like complement the activities of the antibodies and phagocytic cells. Okay. Now, which of the following serves as a defense mechanism in adaptive immunity? Select all that apply. Basically, it is asking you what are the components of adaptive immunity. What are the components of adaptive immunity? We just discussed, like,
Is B and D.
yes. Okay. It is B and T cells that are the main components of adaptive immunity. Okay. Now, this is the match the following that is based on the process of wound healing. Okay. It is already discussed in prior weeks. So it just since we are talking about skin tissue engineering and mostly whenever people talks about wound healing process, they generally talk about wound healing over skin because it's easier to visualize wound healing. And also whenever you're transplanting any device, maybe uh, to the root of skin, like through injection or subdermal or transdermal kind of transplantation, then most obvious place where the injury will be, it will be on skin. So that's why we are discussing it again. So this is the like match the following. If you are familiar with the process, feel free to give it a try.
Okay. Hemostasis will be related to blood coagulation.
Hmm. Yes, yes, you're correct. Next, go
Uh,
on.
Inflammation will be granulation epithelialization.
So inflammation basically talks about immune response. So give it a try again.
Okay.
Leukocytes and macrophages. Yes, you're correct. Next. Mhm. Next, go on. There are two more left. You can be only 50% wrong.
Remodeling will be collagen fiber.
Yes. And proliferation will be this. So I'm just asking, are you familiar with the wound healing process? Or do you want me to explain it a bit? Because I felt like it's not very clear. Yeah. Do you
Brief
want me to?
explanation.
Yeah, yeah. Okay, okay, I got it. So, general wound healing process, these are the four steps, and the sequence is also like this. First is hemostasis, okay, then comes inflammation, then it is proliferation, and lastly, the remodeling. So, this process is discussed in absence of any biomaterial. So, in the previous few minutes ago, I discussed how the wound healing process is modified in presence of a biomaterial. Now, here, biomaterial is not there. So, if any wound is created, our body will respond to any foreign objects like the bacteria or any dust and all that might have entered into our body. Okay. So, first step is hemostasis. Here, a creation of provisional matrix happens, which is nothing but a fibrin matrix. So there are proteins called fibrinogen in the blood plasma that gets cross-linked in the presence of enzyme thrombin, and as a result, fibrin gel kind of a structure forms. Now, the secretion of this thrombin enzyme happens because of the activation of clotting cascades by platelets. So there are various types of clotting cascades that starts. So, and the result of that is the formation of this provisional matrix. Within this matrix, the platelets they get trapped, and as I told you, there's a dilation of blood vessel as well. And because as a result of secretion of chemical mediators by the injured cells. So when that happens, the permeability of blood vessels also increase. So so neutrophils, which are the phagocytic cells, they can also come at the site of the injury and start secreting more and more types of cytokines to call upon more cells. So all these cytokines, then platelets, like injured cells, so they get trapped in this fibrin matrix and results in hemostasis. That is, the blood stops flowing. Okay, and a blood clot gets formed. Now, once this is done, once the barrier is temporarily closed, then the process of inflammation happens. Now, few neutrophils have arrived. Now, it is going to call upon more neutrophils and more types of phagocytic cells like monocytes. These monocytes they differentiate to form macrophages. Now, their role is to phagocytose any bacteria or viral particle, anything that has entered inside your body. Okay, and at the same time it will also secrete more and more cytokines to call upon or to initiate the proliferation and of different types of cells. So in this step, the endothelial cells get proliferated, the fibroblast cells, then if it is skin, then there is keratinocytes as well. So any type of resident cells of the tissue they starts proliferating, and they try to restore the normal structure of the tissue which was there prior to the injury. Okay, so endothelial cells they will try to reform the blood capillaries through the process of angiogenesis. Then fibroblast cells, they will secrete more and more collagen to restore the, say, dermal structure of the skin. Keratinocytes, they will proliferate to restore the epidermal structure of the skin. Okay, and as subsequently you will see that in this step, in the proliferation step, um, what happens? The wound gets closed. So, apart from the normal fibroblast, there is another type of fibroblast that also exists in this step, that is called myofibroblast. So they have the property of both a fibroblast cell and a muscle cell. So they are the primary cells that actually are responsible for pulling the both ends of the wounds closer together. Okay, so they are responsible for wound closure, wound contracture. And all these cells, they proliferate, they're adherent type of cells, right? So they need a substrate. So a granulation tissue also gets formed, onto which these cells proliferate and migrate and fill up the space. Granulation tissue, as I already discussed, if you ever try to, maybe out of curiosity, plucked out the blood clot, it is the very pinkish granular appearance type of tissue that you can see below it. That is a granulation tissue. And over a period of time, you will see that granulation tissue disappears. The keratinocytes and the melanocytes, they all come and fill up that space. Okay, so it's like, and the texture and the color of the area becomes similar to the surrounding area. So that is a result of the proliferation step. Now, once this proliferation is done, virtually the wound is closed, okay? But uh, the inner structure, like the ECM structure, it is still undergoing, um, it is still in a dynamic state. It is not fully formed. So that is when the remodeling step happens. So here, the collagen that was secreted in the proliferation stage by the fibroblast, so they get reorganized. Okay, the fibrils, they get reorganized, they try to create the ECM structure as similar to its original structure. And this remodeling stage, it can go on for years. Okay, months to years, whereas proliferation, it only after proliferation only this remodeling stage will happen. So yeah, this is about wound healing. Okay, this is about wound healing step. And if you have any queries, let me know.
Understood now.
Understood? Okay. Let's move on. So, which of the following are the examples of alarmins? Okay. Um, do you know what are alarmins? Hmm? Are you familiar with this term, alarmins? Hello, am I
They are
on?
removed by
Mhm.
damage or stress levels. Like they act like a danger signal. They kind
Yeah.
of alert the immune system to, in case of any threats from the external environment.
Yes. So you're correct. So they are the danger signals, and they are actually produced by your body's own cells. So when the cell is undergoing necrosis, like not the programmed cell, that not apoptosis, but a program, but through necrosis, when the cell gets damaged, lysed, so at that time, these alarmins get synthesized by the cells. They act to call upon the other immune cells, the phagocytes and all. They will come at the site and clear up the mess. So, which of these followings are those alarmins or the examples of those alarmins? A. ATP, B. Uric acid, C. Heat shock proteins. D. All of the above.
All of the above.
Yes, you're correct. So this is the slide that talks about what are alarmins. So alarmins are endogenous equivalent of pathogen associated molecular patterns. Basically, our body's own antigens. Okay, so they are very similar to the pathogens that comes into our body. Very similar to that. The examples like heat shock proteins, ATP, and uric acid, they are released by cells which are undergoing death by necrosis. And these alarmins, they are meant to call upon cells like macrophages and dendritic cells so that they can come and, as I told you, clear up the mess. So these are the receptors that are present on the macrophages like scavenger receptors, toll-like receptors, C-type lectins, which can recognize these alarmins. Okay. Which of the, sorry, choose the correct step regarding impaired wound healing and excessive fibrosis. So I discussed about the host response to biomaterial. So it is related to that question. It is related to that topic. But here we're talking about impaired wound healing, and the fibrosis has gone on very extensively. So the process goes on similar. Only thing is the intensity is higher. Okay, when the wound healing has impaired. Otherwise, the steps that I told you, those are exactly the same. So you have to recognize the sequence of those steps in the options. A, activation of fibroblast, then FBGC, that is foreign body giant cell, release pro-fibrotic factors in response to IL-4. Then collagen deposition happens. B, first FBGCs releases pro-fibrotic factors in response to IL-4, and then activation of fibroblast will happen, and then collagen deposition will happen. C, first collagen deposition, and then FBGC releases pro-fibrotic factors, and then fibroblast activation happens. And D, first collagen deposition, and then activation of fibroblast, and lastly FBGCs will release pro-fibrotic factors. So which of these options gives the correct sequence um, for excessive fibrosis to happen?
B.
Yes, you are correct. So this is like the diagram that will help you understand what goes on. So this yellow portion is the surface of the biomaterial onto which some proteins from hemostasis step, like the earlier step when the blood and material was interacting, in that step protein got absorbed, this green one. Okay, now since protein is adsorbed, so cells like neutrophils and other type of phagocytic cells, they can easily now adhere onto that surface. So once it does, it will secrete some cytokines, okay, like IL-4, IL-13, IL-6. These cytokines are released, and as it is done, it will also further lead to formation of foreign body giant cells. So these foreign body giant cells, they are basically a result of fusion of these macrophages. Okay, and this happened because of, again, the signals by IL-4 and IL-13. These foreign body giant cells are formed. Now they are responsible for secreting these matrix remodeling proteins, ECM remodeling proteins. These are enzymes, basically, matrix metalloproteases, MMP9. Then these proteins get released, so they will be responsible for remodeling. And in presence of this, and also fibroblast also releases some collagen. So as the collagen gets deposited, it will create fibrotic capsule, But remodeling also takes place at the same time, means the capsule gets much more finer in structure, and new bonds among the fibrils, collagen fibrils get formed. Okay, so yeah. If you have any queries at any point of time, feel free to ask, all right? This is the next question. What is the first event in the host response to an implanted biomaterial? A. Chronic inflammation. B. Fibrous encapsulation. C. Protein adsorption on the surface of biomaterial. And D. Formation of granulation tissue. What is the first event that's going to happen when a biomaterial is implanted
Protein
in your body?
absorption.
Yes, you're correct. This is the next question. Which immune cells they are typically involved in the acute inflammatory response to a biomaterial? A. Macrophages and fibroblasts. B. Polymorphonuclear leukocytes. C. B cells and T cells. And D. Eosinophils and mast cells. So we're talking about the acute inflammatory response that goes on for a relatively shorter period of time. So which type of immune cell is predominant at this stage?
Macrophages and fibroblasts.
Okay. So actually it's B. Okay, the correct answer is B. So I told you the neutrophils, right? So neutrophils, they are the cells that are also called PMNs or polymorphonuclear leukocytes. So they are the most abundant type of phagocytic cells available in our blood. So they are the first one to arrive, and their concentration is the highest when the acute inflammatory response takes place. Okay. Apart from that, other type of phagocytic cells that belongs to this class of PMNs are basophils and eosinophils. Okay, but it is predominantly the neutrophils. But neutrophils, they are very short-lived in nature. Like they will last for only 1 day or 2 days at the site, and that is why it secretes, goes on secreting more and more cytokines to call upon these monocytes and then macrophages. Now these cells, they can stay at the site for months. Okay. The next question, what types of immune cells, what type of immune cells fuse to form the foreign body giant cells around implanted biomaterials? A. Neutrophils. B. Eosinophils. C. T cells. D. Macrophages. Foreign body giant cells, we just talked about it. Remember, in the earlier slides, we talked about it. Yes?
Macrophages.
Yes, macrophages. So, um, and as the information process is going on, uh, against a biomaterial, due to the size of the biomaterial, uh, these macrophages, they will fuse, all right, and become much larger cells, and they are having multiple nucleus and a large amount of cytoplasmic content, a large amount of lysosome vesicles and other organelles. So these cells are predominantly going to release large amount of proteases and ROS in order to degrade the biomaterial, okay, slowly and slowly. And they tend to do it at a very close proximity to the biomaterial. What is the main factor that influences whether a biomaterial will cause a foreign body reaction? A. Material surface chemistry and roughness. B. The biomaterial implant. C. The shape of biomaterial implant. D. The density of immune cells in the host tissue. Which among these options, yes, do you think is important to determine whether the foreign body reaction will happen or not against a biomaterial?
Material surface chemistry and roughness.
Yes. So you already know that cells can only see the surface of the material. So if you can modify the surface, then you can make sure that foreign body reaction happens to a much lesser extent. But if it is not modified well, then foreign body reaction can happen to a very large extent. So it's the surface and the functional rules that are present that is going to be very important for us, as well as the roughness. So if the surface is very smooth, like that of a breast implant, then people have observed in ex vivo analysis that only 1 or 2 layer thick fibroblast cells are there. Okay, but if the surface is like that of a fabric type of scaffold, and on those cases, people have seen a large amount of FBCs, foreign body giant cells, as well as macrophages onto the surface. So depending on how rough or how smooth the surface is, accordingly you will see the density of these foreign body giant cells near the surface. Which process can reduce the intensity of foreign body reaction to a biomaterial? A. Increasing the size of the biomaterial. B. Modifying the surface properties to make it more hydrophilic. C. Using highly immunogenic materials. D. Introducing rough surface topography.
Option B.
Okay, so can you tell me why you thought option B is correct? By the way, is correct, but let me know your thought process. Why do you think if you make it hydrophilic? Yeah, go ahead.
Okay. Mm, Maybe the immunogenic cells might not be attached to the surface of the biomaterial by modifying the surface properties.
Mm-hmm. Okay. Um, so suppose I give you two surfaces. One is very hydrophilic, like maybe the contact angle is like 50 degree. Another surface is hydrophilic, also hydrophilic, and the contact angle is, let's say, 85 degree. Okay, so which surface is going to have the ability to reduce the foreign body reaction? A or B?
B1.
Okay, so why B? So both are hydrophilic, but one is A is more hydrophilic than B because the contact angle is less. Okay. So since you said that, since you think that making more hydrophilic will make the surface much more favorable to reduce the Farn-Body reaction, so then why do you say that choosing option B or choosing the surface type with contact angle 85 degrees is going to help more to reduce the Farn-Body reaction? Why not A? You get my question? Mm-hmm. Yes, go on. Why making the surface hydrophilic is going to be useful? I know among all the four options, it seemed more obvious, but why? We have already discussed this previously as well. So, just a recap of that, we'll do now.
Yeah, maybe to know in order to reduce the protein absorption of the immune cell adhesion so that the biomaterial surface and the immune cell interaction won't be as aggressive as for surfaces that are usually not more hydrophilic.
Um, so you're saying that hydrophilic surfaces will not allow better protein absorption, right?
Yes.
Mhm. So, yeah, you're correct. But why? Why it will not allow protein absorption more? What goes on? Like what goes on the surface that prevents protein absorption on a higher hydrophilic surface?
The bonds are not as strong.
So yeah, let me now give my thoughts into it. So if the surface is very hydrophilic, then water molecules that are there in the blood, so that those water molecules will have or will form much more stronger hydrogen bond with the surface. Okay. So when the protein now comes, now when it tries to, adhere directly with the surface, so the water molecules has to first remove, right? Then only the proteins can come. Now, since it is strongly adhered with the surface, it becomes kind of thermodynamically unfavorable. It is reluctant to go on, to move on. So the degree of proteins that will adsorb on this particular hydrophilic surface is going to be relatively less than the proteins that are going to get adsorbed on this surface. Okay, so now adsorption should also happen in such a way that the structure of the protein should stay intact and all the necessary binding sites should be exposed well so that when the cells come and they try to adhere onto the surface, they find the necessary um, sites to bind to it and adhere. So there is a term called non-fouling surfaces or non-fouling biomaterials. Now these biomaterials basically take help of this principle. Their surfaces are highly hydrophilic, highly hydrophilic. One good example of this kind of material is PEG. And because they are highly hydrophilic, they don't allow protein adsorb on it. So neither our plasma proteins can adsorb, nor any bacterial biofilm can form on it. So that's why they're also called inert biomaterials. So when you are working with this kind of biomaterials, the intensity of foreign body reaction is going to be less, and like they are going to be like stay at the site for a longer period of time, can do the action. Okay, and subsequently, if you have synthesized or if you have delivered nanoparticles, then they will easily go out of your system. Or if you have like transplanted it, so peg is not known for biodegradation, then it may be like it will stay on in your body for few years and not going to cause much trouble. So yeah, so that's why making more hydrophilic, or if you use highly hydrophilic substances, that will reduce the foreign body reaction. But the other way, you may wonder, what if we make it highly hydrophobic? What happens then? So, highly hydrophobic surfaces, the story is like similar but slightly different. So, when the surface is highly hydrophobic, then what happens? It will not allow water hydrogen bonds, right, to get formed with water molecules. So the water molecules, whenever they come in contact with the surface, they are binding with the surface with very weak hydrophobic interactions. Hydrophobic interactions. And now when the proteins, plasma proteins and all, they come in contact with the surface, when they want to now bind, so these water molecules are like, okay, fine, I'm ready to leave. I don't want to stay here any longer, because the bonds are very weak. They are thermodynamically very weak, favorable. That makes the breakage of these bonds, breakage of these interactions, hydrophobic interactions, thermodynamically favorable. So it goes on. These water molecules easily move out. So now these proteins, they are now free to interact with the surface, right? But what happens? These proteins, they uh, while interacting, it often happens these proteins denature. Okay, they lose their structure. They are not their native structure are not the same anymore, because of which, let's say, they deform and denature on the surface. So because of which the active sites for binding, they are now absent. So then also when the cells are trying to come and adhere onto the surface, because of protein onto the protein, they are not able to. Okay, so highly hydrophobic surfaces also, I'm talking about highly hydrophobic surfaces, also do not favor cell adhesion. But protein can very well adsorb on it, but it cannot favor cell adhesion. So that's why people say, in order to achieve good cell adhesion onto your substrate, you have to stay within a optimum range. If this side is highly hydrophilic and this side is highly hydrophobic, then you have to stay like in between. In between is the sweet spot for cell adhesion. And when you stay within the sweet spot, then um you can like you cannot avoid foreign body reaction, because you also want your therapeutic cells to adhere as well. But if you want to reduce it, maybe a little bit shift towards hydrophilicity will help, but then it will compromise the adhesion of your therapeutic cells. So yeah, this way we have to optimize like how much of the reaction we can sustain, whether it is affecting our application or not. Even if so foreign body reaction is happening and your therapeutic effect has completed, then it's fine. It's fine that foreign body reaction should happen, but you cannot completely avoid it. Okay, our body doesn't like our body is a very works tries to work in optimum fashion. It does not have any extremes. So when we are working with these problems, like tissue engineering problems, we also have to find those optimum ranges. Okay, so yeah, this question is clear.
Yes, ma'am.
Yeah, I hope you will not forget it anymore. Okay, which of the following describes a characteristic feature of fibrous encapsulation stage in response to biomaterials? A, it occurs within hours after implantation. B, it is short-term inflammatory response. C, it is composed mainly of collagen and fibroblast. D, it prevents biomaterial degradation. So which of the following options describes the fibrosis stage basically? Mhm. Anyone?
C.
C. Yes, you're correct. We've already discussed it, so I'm not going to go further. But if you have any questions, let me know. Okay. Immune response to degradable materials, degradable implants, is influenced by A. Degradation products, B. Surface changes due to degradation, C. Both A and B, D. None of the above. Which of these options, A, B, C, D? Right. C is like both A and B, D is none of the above, is going to influence the immune response to degradable implants.
Both A and B.
Yes, you're correct. So as the products are released, the byproducts of degradation are released, they are going to, maybe get engulfed by macrophages, or as the surface is getting degraded, it is becoming, um, shallower, then more and more functional groups are going to get exposed. So all this thing will together give a combined immune response. Okay. So which is the regulatory agency that markets medical instruments, sorry, markets nahi, that approves medical instruments and tissue engineered products to reach the market? A. Federal Drug and Development Agency. B. Food and Drug Administration. C. Food and Development Agency. D. Foreign Drug Regulatory Agency. Anyone heard of FDA? So if you know the full form of FDA, that's the correct answer here. B.
Food and Drug Administration.
Yes, 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 correct. So, and this is an organization in USA, and they are like generally look after the food and the drug, but as well as they can approve the bioengineered products like the medical devices and tissue engineered products. In India, there are similar groups as well, the FASI, and I'm not recalling the drug administration name. I think it's called CD. Yeah,
CDCSO.
yeah, yeah.
CDCSO.
Yes,
It's
yes,
the
yeah.
CDCSO, yeah.
Yeah, correct. So these are the organization in India. Okay. The next question: which of the following does a tissue engineered skin requires? Select all that apply. A. Provide inelastic support to skin. B. Promote vascularization. C. Promote or provide a layer for suppressing the growth of keratinocytes. D. Securely attached to the underlying dermis. Which of the following features does a tissue engineered skin should require or must require, you can say, for successful uh, integration with your body?
Yeah, D.
Huh,
Securely
or
attached
like,
to underlying
huh,
dermis.
obviously it should, but there's one more option that is correct here.
Promoting vascularization then?
Yes, you're correct. So these two things, if they happen, they can make sure that your graft is sustained for a longer period of time, as well as if you have used biodegradable biomaterials, so it will allow easy clearance of the biodegraded products from your body, and it will like it will not require any later surgical intervention to remove the scaffold. Okay, so previous generation of skin tissue engineered skins, they were like simply membranes, but it was very difficult for those membranes to get integrated with your body because there was no biological factors. And if they were using the membranes for larger wound, then what happened is like cells couldn't migrate a lot on the surfaces because there were not enough uh, factors like cell binding factors present on the surface. So because of that, they then later added cells to it on, and cells were cultured onto that membrane that is later generation. So now they are like membrane, planar sheet basically, but there are cells on seated or grown on top of it, and then they place it on your wound. Now what happens, now biomaterials and the cell interaction should be such that it is not causing a very extensive immune response first, but it should also allow the migration of the host cells like the endothelial cells, the host fibroblast, host keratinocytes. They should also come and live on that particular or repopulate that particular tissue engineered graft. So once that happens, it can give rise to, or you can see new blood vessels getting formed within the scaffold as a result of angiogenesis. So when that happens, uh, like it is leading more and more towards successful integration into your body, right? So vascularization helps in clearance of any waste as well as regular supply of nutrients. So the host cells that you have provided, those are alive first, and then the sorry, not the host cells, the artificial cells, therapeutic cells that you have created, given outside, as well as the host cells that are now coming, they are also having sufficient nutrients. And when it is successfully attached to dermis, so any dermal secondary structures, they can also now interact with that particular scaffold. And hopefully you can see some secondary apertures like hair follicles or oil glands, they may start forming. Okay, but I have not seen yet successfully happen yet in the literature. But yeah, I have seen people have used very nice combinations of biomaterials and skin, biomaterials and cell types that do promote vascularization after implantation in mice models. So yeah. Now this is the next question. Which type of skin graft will consist of an epidermis and a full dermis layer? A split thickness or a full thickness skin graft? if I tell you what is split thickness and full thickness, then I would like answer it. But I want you guys to give it a try first, maybe a guess, then I'll explain.
Full thickness.
Yes, you're correct. So full thickness grafts basically consist of both layers of the skin. You know, skin consists of only two layers, epidermis and the dermis. The dermis is further supported by a fatty layer that is called hypodermis. So hypodermis is not part of the skin. It is just insulation, you can say, that keeps our skin basically warm, our body warm. So it's like subcutaneous fat that is called hypodermis. And just below the hypodermis are the muscles. So that is full thickness skin graft. Now, if there is no need to um, so if the dermis is intact, only the epidermis is compromised, then we use split thickness skin graft. So split thickness is only the um, like membrane kind of a structure onto which keratinocytes and melanocytes, they are just transplanted, means they are just cultured onto this. That is called epidermis layer, and that graft consisting only epidermal cells, that is called split thickness. Okay, so full thickness, both layers, epidermis, dermis, split thickness, only dermis layer. And depending on the extent of the injury onto your skin, like whether both layers are gone, maybe due to some deep burns, like third-degree burns and all, or some chronic wound, or like diabetic ulcer. So in those kind of cases, you may need a graft, like a full thickness graft you may need. And in second type, if it is like a big injury, but it only compromised the epidermis layer, then a split thickness graft will work. Okay. So this is the last question for today's session. The wound happened at epidermis. Then the process of proliferation happens with the help of which type of cells?
Keratinocytes.
Yes. Keratinocytes, they are the resident cells of the epidermis. If you are familiar with the structure of the epidermis, so it is basically um, around 4 layers, but it is 5 layers on your palm and sole of your feet. So the first layer of the epidermis is called basal layers, a stratum basilae. I'm just writing basilae. It is stratum basilae and it consists of epithelial stem cells, which rarely divides and only divides when it needs to. And apart from these stem cells, there are other um, native form of keratinocytes like progenitor keratinocytes also stay here. Now, once these cells are here, they start proliferating and move upwards, then move upwards towards spinosum stratum, spinosum layer. Now here starts the cell starts getting keratinized, means the morph secretes keratin. Cells start secreting keratin, so they become keratinized, because of which the morphology of the cell also changes. Then it moves further up towards granulosum, stratum granulosum layer. Now here the cells have become flattened. Okay, so also the structure of the cells has changed a little bit, and then and here at this layer stratum granulosum, here also cell starts dying. Okay, so once it reaches to the top layer, which is stratum corneum, here is the layer of dead cells. These are basically cornified and well protected by lipids. That is why our skin is very moisturized, and when it gets dry, you like see white lines on your skin. That is basically your dead skin cells that are coming off. So every 28 days we shed off the topmost layer of our skin, and once that topmost layer is gone, the below layers, they start sending out new cells on the top. So in this way the cell proliferation is happening in a repetitive manner. Okay, and every 28 days you get a fresh layer of skin. So, and if this layer of epidermis, if this is the entire epidermis, and if this gets compromised, then you need keratinocytes. Okay, to like further repopulate that area. So, and like people have seen that in vivo, when you just seed, when you just transplant few layers of cells, they will once in get contact with your blood vessels and the host cells, they start forming this stratified structure of epidermis later on. But I have also seen people stratifying it beforehand, like they stratified the skin layers beforehand within the culture, within in vitro, in lab, and then they transplant. So that is also possible. Okay, so both ways. All right, then I'm done with the questions, but I'm open for questions from you guys. If you have any doubts regarding any of the topics discussed this week or any other week prior, let me know. I'll be happy to help.
I had a rather general doubt. I mean, the exams are coming up in 2 weeks' time. I wanted to ask
Mhm.
how to prepare for them because this is my first time I'll be giving exams for NPTEL courses. So,
Okay,
advice or lessons I need to look for, whatever.
so the questions will be very similar to what is discussed in this problem solving weeks, as well as very similar to assignment questions. So, like, a pattern will be very similar. You won't get any different pattern than that. First, second is the online. It will exam will be online. Okay, so you have to sit in front of a computer, and it is well timed. So make sure to like brush up the theory as much as possible so that you like maybe it's possible the software gets hanged, like you may not be able to go back or something like
Right.
that. Be prepared for those kind of situations because it can happen. I have given NPTEL exam like many years ago. Okay, so it was online for me, and the question type was also very similar to what I have done for the assignments. Just don't get worried too much. Just go through the lectures well. um go through go to the questions, practice these questions. I have solved plenty of questions every week. You can rewatch those, and it will help you memorize the facts and understand the concepts well. So yeah, it's really fine if you do these things. It's like any other exam, so you'll be fine.
Right, got it. And the questions that were discussed in the prior discussions, where can I get those?
So I have uploaded all those in my YouTube channel. So if you can go to your NPTEL login, you will see after you have logged in on your left hand side, there is a tab says problem solving sessions. So if you click on it, you will find an Excel sheet where you will
Right,
see,
yes, yes, got
yeah,
it.
yeah. So you will see
Yeah,
the week wise recordings. Okay. So yeah, all the 6 weeks I have uploaded. Today also I'll upload. So yeah, you'll get the 7th week lectures as well.
got it, got it. Thank you so much. And I also had one technical doubt, if I may ask, like, from the topics. So, this is from the assignment. Is it fine if I ask an assignment question?
Sure, but I cannot give you the right answer. I can just help you solve it. So
Yeah,
yeah, go ahead.
no. So, for the week 7 assignment, the
Mhm.
third question asks about what is the endogenous equivalent of PAMPs, and as far as I know, the answer is supposed to be damp, but the hint says that it is a 8-letter word, which does not make sense to me. So,
I think we discussed it today. We
oh,
discussed this
I
question today.
joined in around 7:30, so I might have missed it.
No problem. Yeah, I can direct you. It is actually the question. So yeah, it is based on this question. Now you figure it
Okay,
out. Okay? Yeah.
all right, got it. Thank you.
Okay, bye bye.
Bye.
Any more doubts? Any more doubts from anyone else?
Now I have a doubt in the week 6 assignment. Actually, it is over. We have in the seventh question, we have a picture question. There's a provided a picture of cell
Hmm?
junction, and we had options denoting ABC, and we were supposed to choose the right option. When I cross-checked it, actually the correct answer is something else, but they've given me incorrect. I gave the correct answer, and it showed me incorrect. So I have a doubt in that question.
So what have you selected?
I selected option B, where A denotes desmosomes, B is hemidesmosomes, and C is the tight junctions. But the
I
correct answer was actually A.
I think you're correct. Okay, so I can ask you to directly get in touch with NPTEL TA, the other guy who like gives these questions and all. I'm just given the responsibility to like conduct these sessions. I don't have anything to do with like uploading these questions on the assignment. I don't do that. But if you go to the Q&A session, okay, you can find there is a guy here who has answered many questions previously.
Okay.
Or maybe you can directly contact Professor Vignesh, right? You can give it a mail, but I think it should be B, option B. I have like I think you're correct. Okay, so like give a mail directly to instructor and he'll know what to do. Actually, I don't know.
Okay.
Otherwise, I would have helped you directly. Yeah, it's Manoj. Is Manoj Kumar, I think. If you see, he has
Okay.
replied. I think he is the instructor. I didn't get in touch. Like I was not given the contact of that person, so otherwise I would have directly. So yeah, I'm sorry.
Okay.
Yeah, I can tell you that you are correct in your thinking.
Okay.
Okay. Mm-hmm.
Yeah. Okay. Thank you so much.
Mm-hmm. Okay. Any other queries from anyone? Okay. Looks like we are done for today, and I'll upload this and you can have access. So maybe by tomorrow morning you can see it. And okay, good night, everyone. Bye bye. I'll see you guys next week. If so, next week going to be the last session for this course. So I would ask you to come prepared with any more doubts that you might have apart from week 8. I'll like as a general pattern, I'll discuss week 8 problems, but I'll be happy to take any other queries from previous weeks as well. So that after that, you will have ample of time to study well, revise, and prepare well for your exams. Okay, bye bye.
Thank you, ma'am.
Yeah.